Okay, the next lab we're going to look at is we're going to look at cellular respiration. This is the process whereby your body takes in food, sugar, glucose in particular, and will break it down to produce ATP. This is going to require the use of your cytosol to begin with in the cell, and then that end product will be moved to your mitochondria. Now we are going to look at fermentation, which is what yeast will do. We're going to use yeast to provide the enzymes for this process, and glucose, which will be the substrate the enzymes will work on. Now different things will affect the enzyme's ability. We have to have enzyme and substrate for it to really work at all, but we also have an optimal temperature and optimal pH for your enzymes to work at. So for the enzymes in yeast, the optimal temperature is 37 degrees, which is body temperature, and the pH is neutral. So we're going to set up 6 different test tubes, and we're going to put them into this test tube. We're going to fill it up all the way with fluid. We'll put some enzyme in, we'll measure it out, some distilled water all the way to the very top, and then we're going to flip this over, and when we flip it over into this glass tube, it will go all the way up, and when you flip it back over, there will be a small bubble, which you can read the bubble. Now that bubble right now is just an air bubble, but when cellular respiration occurs, the end product of yeast enzymes breaking down the glucose will be the production of carbon dioxide. So we'll produce a carbon dioxide bubble. And the bigger the bubble, the more efficient that cellular respiration has occurred. So we're going to alter the different test tubes and have different materials in them. In the first test tube, we're going to just put the yeast. So we're providing just enzymes. And we will see if there's any degree of cellular respiration. In the second test tube, we're going to put the yeast plus the glucose, the substrate. We should see some cellular respiration, so there should be some development of a carbon dioxide bubble. In the third test tube, we are going to put the enzymes, the glucose, and magnesium sulfate, which is a cofactor which helps the enzyme work even better. And those 3 test tubes will all be at 37 degrees centigrade, which is the optimal temperature. So we'll see if there's any development of any kind of CO2 bubble. The 4th test tube, we're going to put the same 3 materials, enzyme, the glucose, and the magnesium sulfate, but we're going to put it at 0 degrees centigrade. So we're checking to see what happens when it's not at optimal temperature. How does that affect bubble production? And the 4th one, we are going to alter the pH. The first 4 were at pH neutral, pH 7, which is the optimal for this yeast. We're going to add hydrochloric acid to the 5th test tube, and that will make the pH highly acidic, and that we'll see how that affects it. And then the last test tube, we're going to use boiled yeast, which has been denatured, so it is non-functional. Now, we've already done the experiment. We've run the hour in the different water baths, and we can now look at our test tubes. In test tubes 1, we started with a bubble. We did not really produce any bigger bubble. Test tubes 4, 5, and 6 also did not produce too much carbon dioxide. Test tube 2, the bubble is a little bit larger. We've lost some fluid down here, so you can see the fluid's been pushed out by the carbon dioxide bubble. And test tube 2 was the one that had the enzyme and the substrate. Test tube 3, we can see the bubble is much larger. This has enzyme, substrate, and it has the cofactor. Test tube 3 and 4, if we compare those two, they have the same materials, but number 3 was at optimal temperature, and number 4 was at 0 degrees centigrade, so that was non-optimal. And test tube 3 and 5 have the same material, but test tube 3 was at optimal pH of 7, and test tube 5 had the hydrochloric acid, which made it acidic. The last test tube had the boiled yeast, so there was no enzymes at all, so it was totally non-functional. So that's the results of these. Now you would have to be able to analyze, measure, you're going to measure the bubble to begin the lab, measure the bubble at the end, subtract the final bubble, take final minus the initial, that gives you bubble height, and if we ran it for an hour and a quarter, or an hour and a half, you would divide by 1.5 for an hour and a half to get the bubble production per hour. Okay? And you need to know how to do that math also.